So the first step in this mechanism is vesicle docking. So we have a G protein which is known as Rab. Now the conversion of this Rab GDP to Rab GTP, it is done by a specific nucleotide exchange factor. This exchange factor induces a conformational change in Rab that enables the Rab to interact with the surface of the transport vesicle. Okay. Actually, this interaction between the Rab GTP and we can say the transport vesicle, this is made successful when the Rab inserts its isoprenoid anchor into the vesicle membrane. Now the initiating requirement of the docking, which is the tethering of Rab GTP to the vesicle, is done. So for the vesicle to be fully docked at the target membrane, the Rab GTP needs its binding partner. So this binding partner is called as Rab effector, which is present on the target membrane. Now the second step is the assembly of the SNARE complexes. Only docking is not sufficient for vesicle to fuse with the target membrane, so the fusion requires other proteins which are called as SNARE proteins. So for the V SNARE protein, we have VAMP, which is present on the vesicle, and for the T SNARE proteins, we have syntaxin and SNAP25. The association or the interaction between the different SNARE proteins form a very stable coiled-coil SNARE complex, which is called as trans-SNARE complex. And from proper fusion between the vesicle and the target membrane, already formed trans-SNARE complex assumes another conformation, and this time it's called as cis-SNARE complex. So when the fusion has completed and the contents which are present in the vesicle have been transported outside the plasma membrane, what next? What we are going to do next? Now it's time for disassembly of the SNARE complexes so that the SNARE proteins can be available for additional fusion events. But the SNARE complexes are tightly coiled by numerous non-covalent intermolecular interactions. So for the dissociation of the SNARE, we require the input of energy, which is mainly provided by the ATP hydrolysis. But there is one more condition. For the ATP hydrolysis to occur, it needs other proteins. Now let's talk about them. So we have a protein which is called as NSF. This is a hexamer of identical subunits. So this NSF associates with the SNARE complex with the help, or we can say the aid of another protein known as alpha SNAP. So the indirectly bound NSF then hydrolyzes the ATP, which releases enough energy required to dissociate SNARE complexes. This was it. Thanks for watching.